Hello everyone and welcome to another video. Today we are going to be talking on extranuclear inheritance. Extranuclear inheritance is the inheritance of non-chromosomal DNA such as mitochondrial and chloroplasmic DNAs. We have some organelles in the cells such as the chloroplast in plant cells and mitochondria in other animal cells that generally have their own DNA, which is independent of the chromosomal DNA. Such DNA are usually referred to as the extranuclear DNA or the non-chromosomal DNA. These DNAs, they code for their own proteins independently of the chromosomal DNA. And the inheritance of such extranuclear DNAs is known as the extranuclear inheritance. So it can either be, it is of three types. It can either be uniparental, biparental, or vegetative segregation. So we'll start with the uniparental extranuclear inheritance, for example, in humans. We look at this, our diagram here below. This is in humans, we have fertilization. Here we have the spermatozoa or spermatozoon, and here we have the ovum. Now, during fertilization, only the head of the spermatozoa usually move into the egg cell. Okay, so when this happens, the tail of the spermatozoa, this area below here, that contains the mitochondria. This is the mitochondria here. That contains the mitochondria is excluded. And because the mitochondria is excluded, it means that the mitochondrial DNA will also be excluded from the offspring that will be produced from the fertilization. So this said, it means that the offspring that will be generated from the fertilization of the sperm, the head of the sperm cell and the egg of the female will only contain the mitochondria of the woman or of the female. Okay, so when this happens, it means that the extranuclear or the non-chromosomal DNA has only been contributed by one of the parents, so it is known as a uniparental extranuclear inheritance. Okay, yes, so all the mitochondria that are usually found in offsprings in humans in an individual is the mitochondria of that person's mother. There is no mitochondria of the father that is usually present in the offspring. Okay. So the second one, we have the biparental extranuclear inheritance, and here the non-chromosomal DNA is contributed by both parents. Therefore, the name biparental. And an example, we have conjugation in fungi. Here we see this diagram below here. Okay, so in fungi, we usually have during sexual reproduction, by conjugation, we have a hypha that is represented as negative, and we have another hypha that is represented as positive. They usually form a conjugation tube and then which grows towards each other. Okay, so where they meet, the head of that conjugated tube will fuse to produce a zygote, and when that happens, the non-chromosomal DNA will fuse together, and this will give rise to the zygote. So you see that here the non-chromosomal DNA is contributed by both parents in the offspring. This zygote will later grow into a zygospores that will produce spores. Okay, so here the non-chromosomal DNA or the extranuclear DNA is contributed by both parents, and thus the name biparental extranuclear inheritance. The third form of extranuclear inheritance is vegetative segregation. Here, an unequal number of non-chromosomal DNA is distributed to each offspring in a random manner. Okay, an example we have budding in or asexual reproduction by budding in yeast. Okay, we look at this figure below. In A here we have a single yeast cell. In B, it starts to divide by budding. Okay, and when it starts to divide by budding, an unequal number of the non-chromosomal DNA will be distributed in a random manner to each of these two cells. Okay, here it moves on to divide to give, gradually dividing to give three cells. Okay, and here the three cells are still being formed, and here the three cells are fully formed. Okay, and so the non-chromosomal DNA is distributed in an unequal or in a random manner to each of these cells. Okay, and this is known as vegetative segregation. So okay guys, that is all that I had for you for extranuclear inheritance. I hope you were able to learn something from that, and we will see again in the next video.